So as I've told you in the previous video, I really wanted to try a cool project. The idea came to me while preparing for the Christmas holidays. Of course, your grandma will cook all kinds of foods and meats and traditional treats. However, what I basically decided was to cut off meats like pork, beef, sheep, goat, and so on. So basically, except chicken and fish, I want to focus exclusively on a full vegetarian diet. Cheese, milk, and egg included, of course. Anyway, grandmas are old people with their own traditions and the general we know what's better attitude. So I specifically asked her, do not cook with pork, beef, sheep, or goat this Christmas. I will only eat chicken this year. She said yes, of course, but I literally found it hard to believe. So the thought crossed my mind that I should science the bejesus out of the issue. So how would I do that? Well, with PCR, of course. So I've started searching online on PCR identification methods of species from different types of meats. So remember, I do have my own portable PCR machine, a portable transilluminator, a portable electrophoresis tank, and all that good stuff. Literally, all the equipment required to test and do all kinds of cool stuff using PCR, I can fit in a small box inside the trunk of my car. So back to searching online. I found a study in which these researchers described a method on how you can, using multiplex PCR, basically simultaneously identify what kinds of meats are in a particular food. So I was basically interested in testing sausages because these are the main culprit. You can mix all kinds of meats in there and not know about it. So after carefully reading that study, the first issue was getting the primers I needed to make this PCR reaction happen. Of course, the primers did not end up coming until Christmas because of holidays delay, I presume, so I did probably end up eating pork last year, but still the project seemed very cool and extremely easy to implement. But boy, I was wrong. After the primers arrived, I put them in storage with the hope that I'll get to working on the project on a later date when I had more time available. And thus came January. For all you non-science people, explaining what a PCR is really beyond the scope of this video. But without getting into too much detail, I'll try to explain the principle of the whole process in the study and that of a PCR reaction very briefly. For you science people, you know who you are, basically 100% of my audience, the link of the study will be in the description below. So the whole principle of the method is the following. First, I would need to extract the DNA from the meat product, like the sausage for example. I would then need to do a multiplex PCR using the primers in this study. So there are 7 primers in total. One primer is common and the rest are extremely specific to chicken, goat, sheep, horse, pig, and beef. The primers target the cytochrome B gene, which is basically common in all aerobic organisms. Cytochrome B is a protein that's found in the membranes of the cells. Now, based on the design of the primers, if any or more types of meats are present in that meat mix, I should be able to identify what kind of meat is present in that mix exactly by the length of the PCR product on an agarose gel. So for example, if I only have chicken in that mix, I should get on the agarose gel a band of approximately 250 base pairs. If I, for example, have a mix of chicken, beef, pork, and sheep, I should get 4 bands, one of 250, 274, 311, and 398 base pairs respectively. So that's the whole basic principle of the method. Extract the DNA, do a PCR with highly specific primers for the cytochrome B gene, put the product on the gel, and see the results. So the first step was to get some meat that I could use for testing. Like any respectable lazy ass would probably do, I couldn't be bothered to go buy proper meat to perform the tests, so I instead chose to steal a small piece of highly processed pork ham from the fridge at the university. You know, stealing is never a good thing, so I made sure I came clean with the owner of that piece of ham after the fact. Anyway, the second step would have been to extract the DNA from the meat. Again, like any lazy ass would probably do, I've decided not to extract the DNA using a proper kit or using well-known lab methods, and instead I chose to do a direct PCR using just two buffers that I developed. For all you sciencey people, a link will be in the description with a recipe and protocol of something extremely similar. Instead of extracting the DNA, I just chose to break open the cells, release the DNA in the solution, and perform the PCR directly from that. I did not extract the DNA like they did in the article. After the DNA so-called extraction, hydrating the primers and mixing the primers in a specific fashion described by the article, it was time to start the PCR reaction. So for the first cycle, I used a 95-degree denaturation step. I've annealed the primers at about 60. And then after the PCR, I finished and placed the product on a 1% agarose gel. The first results spoke for themselves. I got nothing. For troubleshooting the issue, I thought of things that may have gone wrong. So multiple things could have gone wrong, but the most likely culprits would have been 1. The primers, 2. The PCR reaction itself, and by that I mean the wrong temperature and wrong times for the PCR steps, or maybe the DNA extraction itself. After looking at the study in more detail, I've noticed that they couldn't PCR meat that was cooked at 120 degrees Celsius. So I also presumed that the highly processed meat was not the ideal source of quality DNA for the PCR. Instead of trying once again, I decided to step up my game and get some proper meat for testing. So that's what I did. I first went to a grocery store that had a butcher store. I asked the lady at the counter for raw meat. I said that it doesn't matter what it is, but I needed some raw meat. When she asked me how much I wanted, I said that just a couple of grams for each would be enough. She looked at me very strange, of course, and asked me what do I need the meat for, since it obviously wasn't for eating. I tried to explain to her that I'm studying for my doctorate, I want to make a cool project in which I want to find out what particular types of meats are in meat produce and so on. Of course, this was getting a bit worrying for them, since she probably thought I was there to test their meat specifically and that I could get them in real trouble. But after explaining further, she didn't seem so worried, but rather intrigued by the idea. The kind lady cut me off some small pieces of meat. I got pork, beef, and sheep, and that's what they had, and also let me have them for free, so that's always much appreciated. I doubt the lady understood what I was doing, since before I left, she asked me on advice on a relative she has that was in the hospital with some health issues a couple of weeks back and didn't get better. I explained to her that I wasn't that kind of doctor, and if she doesn't trust what they told her at the hospital, maybe she should get a second opinion from a healthcare professional. That always helps, and although I don't give medical advice, this would be a good idea. Anyway, so I got the meat for free, and now I was off for the lab to start testing again. Of course, I needed to make some changes in the protocol, so I decided to switch the annealing temperature of the primers, since maybe the primers don't bind at such a high temperature, to up the denaturation step to 99 degrees Celsius, since I thought this would help with the PCR, and also to switch from the 1% agarose gel to a 4% one, since this would give me a way better separation of the small bands on the gel. I also used the meat I got and decided to put a small piece of each in a 1.5 milliliter tube and extract the DNA like I did before. So I've eyeballed some small pieces of meat, cut them in very small pieces, boiled them for 10 minutes, added the second buffer, and from that I took 1 microliter and used it in the PCR. I've tried this like 5 times because I was still having issues. I've tried mixing the meats in every way possible, I've tried changing the steps, I've tried putting less meat in the reaction, I've even tried an in-house magnetic bead DNA extraction protocol. However, nothing seemed to work as it should. I did get banned sometimes I didn't, sometimes the thing I knew worked well all of a sudden didn't. This was getting really frustrating. I honestly did not think that I'll be able to finish the project like I wanted to. One thing in common though was the sheep. for some reason, anything that had sheep or had sheep mixed in did not want to PCR, and of course I had no PCR product. So I've decided to try this one last time. It was back to the grocery store from there. It was literally my fifth time in the grocery store in the past 2 weeks asking for unusual small quantities of meats. So the first time the meat was free, but the second, third, and fourth time, because, I felt kind of bad, I also bought some cheese. The cheese was actually really good. Anyway, I'm pretty sure by now the lady and the actual butcher thought I was either crazy or just stupid. First thing they asked me was if the tests are going as they should. I was honest with them and told them that the problem could be the sheep. Maybe the sheep interferes with my reaction and doesn't appear in the test somehow. But luckily, the butcher had a possible reason on why that was the case. He knew for a fact that goats don't get cancer, and that goat milk had anti-cancer properties. I was obviously very skeptical since goats and sheep and humans are mammals, not to mention that goats and sheep are very closely related, both being in the family Caprinae. As an unrelated subject, I did decide to closely look into that, and as I presumed, in fact, both goats and sheep do get cancer, so sadly, no Nobel Prize for me anytime soon. However, maybe I could do the next best thing and save this project. So I thanked the butcher and the lady and set off with my free sausages. One was beef, one was pork and beef, and the last one was a mix of pork, beef, and sheep. So for the last test, I've decided that the problem was the extraction itself. To be more specific, I thought that I was putting way too much meat in my tubes prior to boiling and adding the second buffer. So I got my free sausages and a piece of chicken breast to act as a positive control. I used a mortar and pestle to grind the meat very very well, and I also added about 1 or 200 microliters of buffer 1. After grinding, I've used a Q-tip with a cotton head just to take a very small amount of grinded meat, just to cover the Q-tip a bit. And then I added that into a tube that had 300 microliters of buffer 1. I mixed it well, I let it boil at 98 degrees Celsius for about 10 minutes, and I then added 300 microliters of buffer 2. I used 1 microliter of that to act as a template for my PCR. Everything else remained the same. I've also decided to spike the free sausages with some chicken meat. So after the PCR, I basically made a 4% agarose gel, and I migrated the free sausages and the chicken control next to some DNA ladders. And the results were exactly what I expected. The first sample migrated at 227 base pairs as chicken should. The second sample with pork and beef, spiked with chicken of course, showed 3 lines at 227, 274, and 398, exactly as it should. The third sample had chicken and pork, and exactly as before, the agarose gel showed 2 bands that migrated exactly as they should, and number 4, the most complicated one, showed 4 bands, each corresponding to sheep, beef, pork, and of course the spiked chicken. So, I finally made this happen. Of course, I could still optimize the reaction and also try other meats, but just for fun, this was enough. So, Granny, watch out what you put in that Easter sausages this year, cause I'm coming prepared this time.